sharing is spelled. And completely opened it. There we go. All right, everybody can see that. Everybody can hear. Oh, sorry you didn't get the massive snowstorm to get to stay home today. All right, so for those that are on Zoom, we're on 657 page out of our big group of this. We're at the apicomplexia, intestinal and tissue coccidia. So we told earlier we do have some good ones today, some you've heard of, some that you might not have really heard of, but you know what the cause of. We'll bring a CDC website up for that when we get there. So the apicomplexia are unicellular spore forming. So we kind of got to introduce some new names, a little different life cycle. But in the feces itself, it's going to look quite similar. In fact, if I looked at one of the pictures real close today, might have seen it yesterday during lab that I was looking hard, and it definitely has the look of one of the ones I saw yesterday. So these organisms are obligate endoparasites of animals. Phylum is diverse organisms such as Coccidia, Hemosporidia, Plasmodia. The apicomplexia comprise a bulk of what is called the sporozoa. So keep that in mind as a group of the parasitic protozoans. These are without flagella, without cilia, and without pseudopods. So the first one we'll start with is Cystoisosporobelly. That was pronunciated for spelling purposes. Sea belly. Worldwide, so it's going to ring true as far as most of the things that we talk about. With these, life cycle is in two stages, two development life cycles. So kind of got a new definition here of asexual reproduction. I think we've mentioned that in sexual reproduction, but we may not have introduced these two words, which is schizogony and sporogony. Schizos occur within the epithelial cell of the intestine, and they allow the spread through the bowel. The sporogony, this is where we get a male and female gamete develops, and this is how we're going to produce the new oocysts, which are going to be passed. And that's where you come in. You come in as far as, what is in the feces? And then hopefully we'll see the sporogony. So there's immature oocyst. I want to say oocyst. I just say that all the time, but you can say oocyst. You can say oocyst. I think it's find it different ways. Ellipsoid, meaning not, circular, kind of got an ellipsoid, if you remember geometry. Ellipsoid shape, spindle shape, contain a single undifferentiated spherical mass, protoplasma. Spherical mass undergoes 2 cell divisions, producing 2 sporoblasts. So we're going to have kind of numbers of development as we look at these. As they develop, we're going to have different numbers show up at different stages. Cell division continues within the sporoblast to produce the sporocyst, which contains 4 sporozoites. Y'all got that already, right? On that developmental stage. So it's just one of those that you just have to draw a life cycle arrow as we develop through this. And this is where we come up with our CDC life cycle for Cystoisospora. We'll take the top because we'll see this is where we come in. So I always like to start here at the diagnostic phase with the oocyst and the feces being shed. And this is what we're going to be seeing first off, right? So that being said, the oocyst, we say we have 1, so we'll keep clicking back, right? We'll keep click back here. So we say that there is a oocyst or ellipsoid spindle shape containing a single undifferentiated spherical mass or a protoplasm. That's here. Okay, single mass, the oocyst, and then it goes through its development. So the oocyst comes out in the feces. The immature oocyst makes sporoblast. So there's two, right? Spherical mass undergoes cell division, producing two sporoblasts. Then that matures more, right? That immature oocyst with sporocyst. We went from blast to cyst. Cell division continues within the sporoblast to produce a sporocyst, which contains 4 sporozoites. So let me get to my picture again, and we'll see this again. So here, immature oocyst with sporocyst goes into the mature oocyst with sporozoites. How many is there? All right, oops, there we go. Cell division continues to the sporoblast, to the sporocyst, which contains 4 sporozoites. So each cyst here, right, contains 4 sporozoites. And that is then infected stage and gets eaten or taken back in to invade the intestine once again. So what happens in the intestine is in the bottom box. So you see in the intestine, the invasion of the intestine. So here is the mature oocyst that we ingested here. And as you can see, 1, 2, 3, 4 sporozoites then hatch from, right, the oocyst. Each sporocyst has 4 sporozoites inside. That would be 8 in each oocyst, right? So as they invade the intestinal wall here, they can go undergo asexual development, right? And then they continue through the intestine here until they produce what is known as merozoites. And the merozoites look a lot like the sporozoites, okay? It depends on where they are in the developmental stage. And those stay in the intestine, and actually one can make a fertilization here, right? Can happen between the microgamete and the macrogamete. One looks like male, one looks like female egg. There's sexual reproduction that goes on to produce what is shed in the feces, which is the oocyst in the feces. That's where we are here. I'll pause for you to gather that info. So I know it looks real simple here, okay, in words. And it looks real simple here, but when you dissect this development, it's not as simple as it appears. And it is definitely a word train for you for test purposes, okay? You see multiple choice questions with these words in different areas in different ways. You need to understand the life cycle. So we'll focus more on the life cycle here than we did previously. Cytoisospora belli, the infective stage which is shed in the feces, is the mature oocyst, which contains the sporozoites. Diagnostic stage we're pretty familiar with. Either the immature or the mature oocyst are being shed in the feces. Morphology, ellipsoid, we've already established. Size-wise, 30 by 12 micrometers. A lot bigger than that red blood cell we've used as our reference. What does it cause? Once it's in the host, disease destructive to the villi, leading to malabsorption, leading to a loose, pale yellow, foul-smelling stool. Have you, let's put it this way, how many of you have smelled C. diff stool? Not yet? C. diff stool is foul smelling. It's a very key to that diarrhea for C. diff toxin. It is foul. Same way here. That would be definitely for you to key in on. Usually the nurse and the doctor have already keyed in on that, as we have a foul smelling diarrhea in the patient's bed, or in their bedside, or in their toilet. So how do we find it? We've been working with this direct examination. Here though, we can add a little bit. We can add an acid-fast stain during this. Aramine rhodamine fluorescent stain to help us. We're going to get to a point where we're going to do a flotation. So get ready for that. We're going to see that as we get bigger with the eggs. We'll do flotation with them. So we're working through direct exam with y'all doing these wet preps and iodine stain preps. We'll move from that, hopefully, once you get keyed in on finding things. You did really well yesterday finding some surprises, hopefully. Some of you found the surprises better than you did the actual keyed in sample, the tropes. We were having trouble with the trophozoites pretty much all day. But you can do a concentration procedure. We have that procedure in your first handout I think I gave you. The oocysts are very light. They float, so you got to exam a focal plane directly below the cover slip carefully to reduce illumination. So again, not always the easiest things to find and see, but we're getting there. Here is in the intestinal stage. So intestinal biopsy, which we will not have. It'd be great if we did and had it, stained with hematoxylin and eosin stain for our prep. But definitely the invasion here. Again, invasion of the intestine. This is more what we will see with a direct wet mount. You're like, oh my God, great. It's flashback time, right? You're already, starting to have flashbacks from going through wet preps. Yeah. You're hanging in there, right? Because when y'all throw in the towel and give up, then I got to go to a video. We're gonna keep on direct wet mounts as long as we're good. Like I said, we're in lecture 2 in lab, so we'll eventually catch up. But just so you know, Giardia is coming. So you're not too far away from Giardia, but there are a few more samples to go before you get to Giardia. This is our direct wet mount. Hopefully you key in on the middle of the slide. You can definitely see that it's a elliptoid shape, football shape. UV light with the fluorescent, you're like, well, why can't we beautify ours like that? Why can't we get a fluorescent microscope that helps us out? All right, questions on C. Valley? Is that our first one? Yep. C. belly. All right. Now we move to Sarcocystis. Man here is the incidental host for the species which infects other animals. Man is the definitive or native host for 2 species. So Sarcocystis has got a variety of different species, okay? We're only the definitive host in 2, and that's Hominis and Sui hominis. As all other Sarcocystis were incidental hosts. How do we become incidental hosts? Just by chance. What's our way we become the incidental host? We're not in the normal life cycle, right? Of the going from one to the other. We just kind of get put in that by picking up the host, maybe ingesting the host, or maybe ingesting outside the host if it's able to stay outside the cyst. The life cycle in the definitive host, male and female gametes develop in the small intestine, producing that oocyst that's needed to be passed into feces to continue the life cycle. And the life cycle is completed when the oocyst is ingested by a new definitive host. When ingested by an incidental host, the parasite still invades the intestinal wall and invades the muscle. That's going to be it, okay? Doesn't get to continue on. So that's what an incidental host does. Just it picks up the parasite, but it doesn't allow for the life cycle to continue. Elongated centrical bodies in the radial striations membrane within the muscle. So we get a sarcocystis invasion of muscle. Okay. You can see that right there. Invasion's hard. See, so sarcocystis, we do direct wet mount or concentrated technique, and we're looking for the oocysts, which are shed in the feces. They're fully developed if we're the definitive host, and the sporocysts rupture out of the oocyst, giving us sporocysts, single or in pairs. And inside those sporocysts, we have sporophore sporozoites within each sporocyst. So again, we could be a single sporocyst, we could be paired sporocyst inside the oocyst, the oocyst, and inside that we have 4. So 4 to 8 sporozoites within each oocyst. That's straight. That's gonna be hard. There we are, and this. Anybody think they saw one of these yesterday? Possibly, right? Possibly, because I'm not ruling out anything anymore, because we really got after it yesterday and found some variety in those feces samples. So that's normal for our specimens. In the last year's class, we did have multiple positives in each specimen. So you would know what you were looking at. I mean, I had a list of things to kind of help guide us through that. But yesterday I thought was really neat because I don't have that list. I'm not going to build a list like that. What I'm wanting you to do is say, this looks fishy, this looks important, and work on IDing that the best we can at this point. But I may have seen one of these yesterday, not sure. We're kind of leaning towards Giardia cyst, just because we were kind of in that same family. But definitely this hook tail inside, did see something similar to that yesterday. All right, so 2 in. How are we doing on time? Good. So Cryptosporidium, worldwide distribution. We have Cryptosporidium hominis, which is only for humans. Cryptosporidium parvum, which is humans and animals. Again, you see that it was previously confined to domestic and wild animals, but increasingly we're seeing it in immunocompromised humans. Cryptosporidium. The life cycle begins with ingestion of contaminated food or water containing the oocysts with sporozoites. Sporozoites are released in the small intestine where they undergo 2 cycles of asexual multiplication within that epithelial. The development of the sexual stage, gametes, follows, leading to the formation of the oocyst, which we need, containing sporozoites, which are shed in the feces, looking for their next victim. Disease, severe watery diarrhea. We demonstrate it as a report as Cryptosporidium. As 2 species, we just can't say one or the other. Okay, we can't go back and say, we know we have hominis or we know we have parvum. We just got to say we have Cryptosporidium present because we can't distinguish the 2. Intestinal biopsy for the schizonts, which are 4 to 8 merozoites invading that intestinal wall. and or we can do the duodenal string test, which we take for the oocyst, where we find those. Direct smear, modified acid fast, aramine, rhodamine, fluorescent stain, or we can do the concentration with formalin ethyl acetate. And then this is the one we're going to get to here. This is Sheather's sugar flotation. And I think one of the things I gave you somewhere, I know in the lab handouts or in the Blackboard shell, we have a sugar flotation recipe, and that's what we're going to work on. I wish we could just all gather at the Humane Society and work with the vet there as you know, all 20 of us or whatever, when they do, because every animal I think that has to go out of the Humane Society has to be clear of intestinal parasites. So if they have diarrhea, they have to be checked, and they use the sugar flotation to look for the eggs. And a lot of those we'll see later on coming, which are going to be larger. The whipworm, I've started seeing some of those. They're easier to find in the feces, even on direct or wet mount or iodine stain. Oh goodness, yes, doesn't that look fun? Cryptosporidium, oocyst, direct wet mount. You know, I think you're pretty keen on finding these now. I think most of you have located a cyst and said, hey, come look. Is that a cyst? You know, just hopefully you got that down. I think we're getting there. Here with the modified acid fast stain helps us out, showing that pickup of the red. Another modified acid fast stain. So Cryptosporidium iosus, immunodetection is another way of detecting it. So direct wet mount, that's what we've been playing with. We also have immunology playing back in here, where we can do an immunodetection for the antigens. Okay, so we actually take the stool specimen, use a monoclonal antibody-based direct fluorescent assay. It's the current test of choice for diagnosis of cryptosporidiosis, and it provides an increased sensitivity over the modified acid-fast staining technique. So I think the question yesterday came up in one of the lab sections is, why is the wet mount? What are we doing? What is the official entry into our result of the direct wet mount? And the direct wet mount is a screen, okay? So when we're screening these species, we're screening to see if we see something abnormal. We think we see a cyst. We think we see a trope, okay? Then there are steps past that where we go ahead and make a trichrome stain official mount, stain it, have it reviewed by our, our pathologist or our infectious parasitic reviewer or that. So it has to go through, it's not just, hey, we're going to screen a stool on a direct wet mount and then we're going to call it and say you need to treat this patient for parasitic infection. There are steps past the screen. This would be one. If we suspected Cryptosporidium, we would actually go to the direct fluorescent assay. So there's commercial products available. See, we got a direct fluorescent assay, indirect fluorescent assay, enzyme immunoassay, rapid test in the U.S. to diagnose cryptosporidiosis. It's 99% sensitivity, 100% specific. Pretty good, right? Not gonna get much better than that ever, right? Unless it's 100. Identifies the oocyst in a concentrated or unconcentrated fecal sample by using fluorescent isothiocyanate labeled monoclonal antibodies. So I think everybody in here, maybe one exception, has been through immunology or has checked the immunology box. That shouldn't be foreign to you, this terminology. It is. You've got a chance to go look it up. Cryptosporidio species, a combined direct fluorescent antibody test for the simultaneous detection of two things, Cryptosporidium oocyst and Giardia cyst is available. Blastocystis hominis. Continue. Again, spherical form here, not ellipsoid. Going to be round, right? 5 to 30. Large central fluid-filled vacuole. Okay, so here is going to be maybe our, what we built on in the first unit, which is that in the cyst, that glycogen bubble. This is just a large central fluid-filled vacuole surrounded by a thin strip of cytoplasm containing multiple nuclei. Approximately 10 different strains of blastocystis, 50-50 on whether they're pathogenic or not. Different strains cannot be based, cannot be differentiated. I think you've pretty much gotten used to that terminology. And here's what I'm talking about, this fluid-filled sac may be confused with who our glycogen in the cyst from our protozoans. All right, not much there on Blastocystis hominis, but it does have an identifier. Here is one, we're going to get into a couple here in the closing part of lecture. This is one. Has anybody ever heard of this? Cyclospora cayetanensis? Anybody? Anybody? Cyclospora. Before we even do the info, we're gonna stop the share here. We're going to do a little, as we like to do, Google search, right? So this is Cyclospora. Oh, it comes up pretty quick. Like this right here. What I'm going to do is ask it for an outbreak of 2020, because this is last summer. Okay. September 2020. Okay. That's from the CDC. Food outbreaks. Food I was looking for. Okay, so this is travel, but I'm looking for the food outbreaks. Let's see. Oh, here it is. That's the one I was looking for. There we go. You remember this? The bag salad mix? Recall? CDC put out a public health warning and said, hey, this was just in September. Here is your state outbreak. We had 38 hospitalizations, no deaths, covered 14 states, 700 confirmed cases. And this is who we're working with today, which is Cyclospora. And so definitely a ongoing public health concern. I know you're thinking, well, it didn't have Arkansas in there, but it did have Missouri. Okay. Started in May, it lasted till July. People range from the age of 11 to 92. And the bag salad. So this one, this came from Streamwood, Illinois, Fresh Express production facility. So it definitely, it's not one where in our notes, and sorry I didn't share that with Zoom. Sorry, I guess I could have gone back to. Okay. So those that, Rachel and Brandy on Zoom, you see your names, but that was just, we looked up cyclospora CDC outbreaks of 2020. If you were wondering what I was doing. So worldwide distribution affects a wide range of vertebrates, including reptiles, rodents, and insectivores. Transmission, the reason I'm updating that with your 2020 outbreak is because the slides have, you know, a historical outbreak. Water and food. How many do we have again in the 2020 July outbreak? How many patients? 700. Remember, 38 were hospitalized. Here in 1996, we had 1,400 people in the United States and Canada traced to raspberries from Guatemala. 1997, another 1,400 people were infected through raspberries and lettuce and possibly basil. So, so what is it? Why is it in lettuce? Why are we getting it infected? So it's really similar life cycle to the cystoisospora, so the cyclospora. Invades the small intestine, prolonged watery diarrhea. In the saline wet mount, we get a spherical oocyst of 8 to 10. Okay, now we're getting back down to who? Getting back down to our red blood cell size. When first passed, the oocyst is unsporulated with greenish morula. And a morula, it looks like you've got like a seed pod. That's what I think of when I hear a morula. Containing 6 to 9 refractile globules containing those 2 sporocysts, each containing 2 crescent sporozoites. Not 4, 2. Iodine wet mount, we see those internal globules stain brown. Modified acid fast, staining is variable, appearing unstained pink, dark red. Here we are with the modified acid fast. Modified acid, and morula looks like a seed pod, right? You got a couple of seeds in there. Okay, cyclospora. And then again, you know, anybody immunocompromised set up. So then we've got our last one here. Is our Toxoplasma gondii. Remember that from immunology? Didn't we talk about this one? Or was that in micro? Where did we talk about Toxoplasma gondii? Preston, tell us, where did we talk about Toxoplasma gondii?
I don't know.
Yeah, we must have talked about it in micro. That's why I call it on you. Sorry. Again, this is the cat, right? And this one's not a big deal unless you're what? Pregnant and changing your cat litter. Kind of give you a hint on where we got this one from. Worldwide, cat family is the definitive host. So what is the life cycle? The schizonts and the gametocytes develop in the small intestine of the cat. Cat feces has the oocyst, contain 2 sporocysts, which each contain 4 sporozoites. If any animal outside the cat family, an intermediate host, ingests the oocyst, and I'll just give you hints to this. If my cat pooped in the yard, the dog may go out there and eat it. I don't know if you have a dog that likes to eat poop. Sometimes mine do. And that's how that would happen. Or any other animal may decide that's a good meal for them and eat the cat poop. And then we have it transferred to another animal. The sporozoites invade the intestinal wall of that animal. It could invade heart, lungs, lymph, or central nervous system. Crescent-shaped organisms known as the tachyzoites multiply, rupture the cell, and spread through the tissue. Okay, and keep that in mind with the tachy. So we're going to see Brady here in a little bit. You all had med term, so you should know what those prefixes mean. Eventually, the cysts are formed around the organism unless the host dies from a massive infection. the host tissue will be eaten by the cat, and the organisms are released to start the cycle again in the cat's intestinal tract. So the cat is the definitive host. The cat has to get to the cat. Okay? So if you have a cat like mine that likes to eat all kinds of different critters, definitely you can see how a cat would pick up the oocyst again. So here's that trophozoite form, the Tachy and the Brady. The Tachy's actively dividing stage destroys the tissue. The Brady, being slow, that's the prefix, means that it becomes dormant, insists within the tissue of an intermediate host. Okay, so you got two forms of the trophozoite. Here, this is an AIDS patient where Toxoplasma gondii has invaded heart muscle. So here's our life cycle of toxoplasmosis, Toxoplasma gondii. So we have our kitty cat up there at number 1. Okay. And as you see, the kitty cat feces can be taken up by a mouse, a sheep, a pig, and it becomes encysted into that tissue. Okay. So that tissue then becomes what? A meal for others. Okay. That would be the infectious stage. It would be infecting the human. Or you could have the cat feces with the oocyst being shed in the cat feces actually infect the human. And as you see, we have the female with the in utero, and that's where we worry the most. Diagnostic stage, serological diagnosis, direct exam of the parasite from peripheral blood, amniotic fluid, or tissue section. Okay, so not going to be what? Unlike our others, not going to be what? Not in stool. Thank you. All right, serum, cerebral spinal fluid is the diagnostic stage. That's different. First one we got, right? One that's different that way. Both the oocyst and tissue cyst transform into tachyzoite shortly after the ingestion. Tachy's localize in the neural and muscular tissue, develop into tissue cysts, the bradyszoites. Those are those ones that stay dormant. If a pregnant woman becomes infected, the tachyzoites, those are the fast ones, can infect the fetus by the bloodstream. Most of us are asymptomatic with Toxoplasma gondii. Generalized infection associated with mild symptoms to the infections similar to mono. Acute infection is severe form with invasion of the internal organs, so immunocompromised or baby in utero. Intrauterine infection, this is a non-immune mom. So it might be good that you have have potential mom, go change the litter box for a while now. But if mom becomes infected early in life and has an immunity to it, she's not going to get infected probably during, but I wouldn't want to risk that, right? The non-immune mom are severe and results in damage of the central nervous system of the fetus. So mom gets infected while pregnant. These infections lead to blindness, hearing loss, learning disabilities, miscarriage, and stillbirth of the unborn. We have immunocompromised hosts, which is very similar to our discussion with all the things that we talked about in immunology. Suffer lesions in the central nervous system, a severe life-threatening infection. And demonstration is serological test, immunofluorescent assay, antibody against Toxoplasma gondii. Questions? All right. No questions? Zoom, you have any questions? Okay, so challenge is to get on top of the verbiage, the connecting arrows, line arrows to to the life cycle so you understand what you'll be looking at when you look at direct wet mount. And then when you see test questions related to info. No questions from the gallery? Good? All right. Well, you guys have a good day. We'll see you tomorrow in lab, not today. And again, we'll stop the